Hi, I'm Sean Mooney, professor at the University of Washington and Chief Research Information Officer of UW Medicine. Over the past 2 years, we have been creating an initiative in medical data science, and as part of that, we have created this seminar. Speakers will include world-leading scientists who will tell us about how data can impact human health and their healthcare. Please use the link below to register for our mailing list to hear about future events in this series. We hope you enjoy them.
Jeff is Chief Data Officer, Vice President, and J. Orrin Edson Foundation Chair of Biostatistics at the Fred Hutchinson Cancer Center. Previously, he was a professor of biostatistics and oncology at the Johns Hopkins Bloomberg School of Public Health and co-director of the Johns Hopkins Data Science Lab. His group develops statistical methods, software, data resources, and data analyses that help people make sense of massive-scale genomic and biomedical data. As the co-director of the Johns Hopkins Data Science Lab, he helped to develop massive online programs that have enrolled more than 8 million individuals and partnered with community-based nonprofits to use data science education for economic and public health development. He is a fellow of the American Statistical Association and a recipient of the Mortimer Spiegelman Award and Committee of Presidents of Statistical Society's Presidential Award.
Yeah, I'm very excited to be able to speak to you, and I hope this won't be the last time that we interact. So I have just moved back to Seattle. I was in Johns Hopkins for the last 12 or 15 years and have come back to take this new job as the Chief Data Officer and also a professor in the Biostatistics program. And I hope that we'll have lots of interactions with the Medical Data Science Group up at the UW. And so I'm very excited to be here to talk to you today. My enthusiasm could not be higher to be able to talk to all of you and interact with you in the future. I'm going to be talking today mostly about population scale transcriptomics, but I also wanted to make sure I didn't forget to tell you about some of the things that we're up to on the data science side over at the Fred Hutch. We've started something we're calling the Fred Hutch Data Science Lab. I got tired of saying the Office of the Chief Data Officer at the Fred Hutch, so we called it the Data Science Lab, or DASL for short. Everything we're doing is up at www.hutchdatascience.org. And then we also have a Monday morning data science newsletter that we send out every Monday that has links about everything we're doing at the Hutch and all of the jobs that we have available and things like that, as well as just interesting data and AI links from around the internet. So if you want a little bit of data science content to make your Monday mornings happy, please feel free to join up and listen into what we're doing there. And there's going to be a lot more about what we're up to. And I think a lot of it intersects with work that you all are doing there as well in terms of clinical data integration with genomics and other things. I'm not going to be talking about too much about that here today, but we should definitely interact about that going forward, because there's going to be a lot of mutual interests, I think, as we work on those kinds of projects. I posted the link in the talk chat, but you can find any of my talk slides are all up at this website. I tend to go pretty fast and have a lot of slides. So if you sort of lose track, feel free to follow along with the slides that you have. So an alternative title for my talk today could be a Precision Oncology, a Tour of Data Plumbing Challenges. And I think this is reflective of basically any big data science project, whether that's something that's an internal merging of electronic health record data and mapping it onto OMOP and then connecting it to genetic data. That's a massive data plumbing challenge. It's a similar thing with massive scale online public data. And so I'm going to be talking a lot about, the sink doesn't fix itself. Someone has to go in and do all of that low level processing and data analysis to kind of get the data together and get them organized. And so you're going to hear a little bit about our journey of doing this, mostly for transcriptomic data today. But the same kind of principles really apply for all of these big scale data science projects or data cleaning projects. To sort of motivate the talk, I'm going to use a really simple example. So imagine that you're a oncologist and you're interested in the molecular measurements that might predict or be prognostic of things like metastasis or other sort of conditions. So a very simple question you might ask is, what gene expression patterns are prognostics of colorectal cancer metastasis? So this seems like a very simple question on the surface, and it turns out to have a massive sort of challenge under the hood. And I'm going to talk a little bit about how we're trying to simplify some of those challenges and make it easier for people to ask and answer questions like this. So we're going to use an example signature here that was a signature that was of interest to some folks that I was collaborating with at Johns Hopkins. It was a 19 gene based risk score classifier for prognosis, again, for the prognosis for metastasis. And there was interest in whether this signature might be something that would be useful for identifying patients at higher risk of metastasis within the clinic at Johns Hopkins. So the question is very simple, the sort of question of what genes are prognostic of colorectal cancer metastasis, but the sort of collection of activities you have to do to answer that question is actually pretty complicated. So you need to start with some data, which you need to have access to about gene expression profiles. Then you need to do some data processing to get those data organized into a way that you can start answering questions. Then you need to be able to do some computing so that you can answer those questions with statistical or machine learning methods. To leverage that computing and to be able to answer those questions with the genomic data, you actually need to take a step back and actually get metadata, which is the labels about those samples. What are the clinical data that are associated with all of the gene expression data that you might have collected? And then ultimately you want to do some analysis to answer the question that you started off with at the beginning, to be able to answer the question, it is or isn't a potential prognostic biomarker. So it's a lot of steps that underlie a really simple question. And so a big focus of my group over the last few years has been trying to simplify the steps in this process so that people can answer these questions much more efficiently and with much less pain. So the question again is, what genes are prognostic for metastasis? And here you're going to see a little bit of a bias from me being a computational person and not having a wet lab. But imagine you wanted to answer a question like this. First, you'd need to find somebody with access to patient samples, specimen samples that you could then use to collect gene expression measurements on. You'd have to collect those patient samples and the information about them. That might take you, say, 6 months or much longer. It often does. And then you'd want to extract DNA or RNA from the samples to be able to do the sequencing. Again, you're seeing a little bit of a bias in the person that doesn't actually have to do this work. And then you need to sequence the samples, which might take another couple of weeks. And then you need to process the sequencing data. And you can see that all the computational steps are very long and all the experimental steps are very short. You can see the bias in who built these slides. So you might process the sequencing data, and it might take you a while to do that in the right way, and then do the data cleaning that you need to do, and then ultimately analyze the data and answer your biological question. And so overall, this might take you 2+ years to be able to do this analysis if you started from scratch without any of the information available, which would be kind of a pain to be able to answer these questions at any kind of speed or any kind of frequency. And so you'll notice that those timelines are a little bit weird, and that's probably because I'm mostly come from the wet lab side or the dry lab side of this, but the point is still holds, which is that the data collection, analysis, and question answering process can take quite a while to complete. And so the question is, are there ways that we can make this process easier, more efficient by doing some of this work in advance? And I think this is relevant for anybody who's working in these areas to think about, how do you create resources that make it easier for people to answer questions more quickly and in the way that they would like to be able to do that? And the cool thing is that we're at a good time to be doing these sorts of problems. I mean, we're at the intersection of two revolutions that are happening at the same time. This, of course, is very intimidating, but it's also very exciting. On the left, you're seeing a plot of how fast or how big really AI models can get and how much compute time we can put into them to be able to make calculations for these large-scale AI models. And you can see that there's been this sort of step change in how fast the models are growing over time. That's largely driven by the access to commodity computing, and then the collection of types of models, transformer models, and others that are scalable to the right scale. And then on the right-hand side, you see how fast, and I'm sure you've seen plots like this. It's true in every type of data collection. This is just true for the genomic data that I most often interact with. It shows sort of how quickly the data is getting cheaper and cheaper to collect. So when I started my career in genomics, it might have cost you, $100 million or $10 million to sequence a single human genome. And now it's, you can do it for $1,000 or less often, especially if you don't have to go to really deep sequencing depths. And so the intersection of data is getting cheaper and there's more of it everywhere. And we have bigger and bigger compute to apply to that data means that there's a lot of change that's happening in our field. So it all feels like it's happening all at once, which is, of course, like I said, very exciting, but also can be a little nerve-wracking as you're sort of trying to keep up with all the different things that are going on at once. As a statistician, that's where my training was. I got a PhD in biostats at University of Washington in the School of Public Health there. And I remember my sample size calculation. So if N is the sample size and you're doing a calculation for a grant, the sample size formula that I remember is, I'm just kidding, this isn't what you actually use, but is definitely maybe a little too close to home, a little too close to comfort, is how many samples you collect is the amount of money you have in a grant or application divided by how much it costs you to collect each sample. And of course, as the number of dollars per sample plummets to the ground over time, the N gets bigger and bigger and bigger. So we have more and more data that are accessible, which allows us to start thinking about how do we analyze data at a population scale instead of in one-off experiments of 1 or 2 samples at a time. So it's exciting to see what the possibilities are as you think about these explosive growth of the data that happens over time. So the predictable thing is actually happening, which is that we're getting more and more data. These are all old graphs. You're seeing this is only up to 2018, but you can see that the amount of data in public sequencing repositories is just exploding over time. It was true back when this graph was current, but it's even more true now that the spectacular growth in sequencing data is not slowed down. In fact, it's sped up, and a lot of people are struggling to keep up with the size and scale of these data. And so can we just AI this data? That you used to see some of these sort of articles about, well, Watson's just going to solve it, or now it's just ChatGPT is going to solve it. And so the question is, do we just shut it all down? Do people that do computational biology, that do computational data science, are we sort of unnecessary now in the era of new AI? And I think my strong hypothesis is no, because there's still a lot of difficult groundwork that needs to be laid in order for any of those technologies to be applicable. So I'm going to talk a little bit about what does that groundwork look like in the context of, high dimensional genomic data. So the good news is that data sharing is improving over time. And in fact, with the new NIH data sharing requirements that have just come out, it promises to be even more data sharing that's happened over time. And so that's really exciting. The downside is that the data aren't always easy to use. Public data are often messy, they're often complicated, they're often not fully annotated, and so they can be a real pain, even though there's a lot of it out there to be able to do useful things with. And so one of the first steps in being able to use any of these data, especially as they're very high dimensional, very complicated, is to be able to process it down into a more usable form to be able to answer questions you might be interested in from the perspective of being able to solve these sort of high dimensional biological questions. So assuming that the data are available online, that somebody's already performed the experiment or close enough to the experiment that you want to perform, you can kind of skip the first several steps in this sort of timeline. And so you've sort of skipped the patient collection and sequencing sample steps if you can find a publicly available data set. The next, though, step is to be able to process that sequencing data, and that can be complicated experimentally, and it can also be, computationally expensive and require the use of very specific and frequently updating computational tools. So I'm going to be talking a lot about RNA, and I'm going to talk about the pre-processing of data that we collect from a sequencing machine. So this is the sort of cartoon guide to the central dogma, right? So everybody has a DNA profile that's unique to them. Pieces of that DNA are transcribed into the mRNA molecules, which are then translated into protein. And so we are interested in this talk and talking about the middle molecule there, the RNA molecules. And we're going to be using sequencing machines to be able to measure that. I used to talk about how advanced this technology is. Now it's ubiquitous. Everybody is doing this sort of sequencing at a large scale. And of course, it's moved on to things like spatial sequencing and single cell sequencing and so forth. But in general, we're going to be talking about the sequencing of RNA for the purposes of this talk. And why are we talking about RNA and not protein? It turns out that sort of scalably measuring protein expression or abundance is still a thing that's remarkably hard to do at any kind of scale. And so mostly we're using RNA as a measure of a surrogate measure for the protein expression levels. But it's also, as we've seen with the COVID vaccines and with the increasing interest in other vaccines based on RNA molecules, RNA is in and of itself an interesting quantity to be measuring over populations of people. And so again, sort of the cartoon guide to how this technology works, we have a genome on the bottom of this plot here. This is one gene that has different components to it, different exons. Those exons can get spliced together. So there's 3 exons in the gene, the yellow, purple, and that's supposed to be orange, but for some reason it's just yellow. Those exons just get spliced together into different transcripts. And what we get is little sequences of characters that come from those different transcripts. And so those sequences of characters might span the junction between an exon and another exon, but they're just sequences of characters that are sort of back to back. And then we have to figure out where they come from and what abundances we get for these different transcripts based on these measurements that we take at a very high throughput scale. And so what this data practically looks like, for those of you, I'm sure a lot of you who are on this call have seen this kind of data before, but the sort of raw data is just billions and billions of lines of little short sequences, C's, T's, A's, and G's, with some quality scores attached to them. That's not very useful if you want to analyze gene expression abundances. You probably can't just slap this into Excel and do something with it. There's a lot of pre-processing that needs to happen before you get a gene expression abundance out of a data set that's sort of so complicated as this one. And so a person who used to spend a lot of time here at the Hutch, Robert Gentleman, who was at Genentech when he made this quote, says, make big data as small as possible, as quickly as possible to enable data sharing, but also, in my opinion, data analysis. So we need to take the big data that we're getting from these 3 GB files per sequencing sample and turn them into something that's much more usable, much smaller, and much more summarized. And the only kind of caveat that I would add to this is we want to be very careful about how we choose what low dimensional summaries we make of the big complicated data. This is true both for complicated data like genomic data, that the primary problem is often size. And it's also true for medical data, whether it's EHR data or other types of data, where the problem often isn't as much one of size, but one of security and privacy. And so thinking about how you take data that is identifiable PHI and turning it into summaries that are usable and analyzable at a large scale is a sort of a similar problem with a different motivating underlying reason for that existence of that problem. And I think it's instructive to think a little bit about what do we mean by the data when we talk about data from a sequencing experiment. So I'm just showing you, all the biochemistry behind sequencing in one slide, which is, to say that I'm not going to go into too much detail. But the way that this works is in the top left top left graphic here, you see little sequences that are stood on end, and these little clusters of sequence all have the exact same DNA sequence. You can sort of grow them on a slide with hundreds of millions of molecular clusters on one slide. And then on the right here, you get what you do is you basically wash this slide with different radio-labeled bases, one color for each base. And then as you wash the slide, you can take a picture, and every cluster will have a different color for each base that gets added to the sequence. And so sort of sequencing by synthesis here, and you get sort of a green dot for this sequence. The first sequence up on the top left corresponds to this little green dot in the image file. And then as you then wash another base on, you get the second letter in the sequence for that little cluster, and the third letter for that cluster, and the fourth letter. And you read it off by looking at this sequence of images on the bottom right. And you look at the same dot across images, and it tells you the bases for that little cluster of sequence reads. And then you turn those into a file like this, where you sort of see for position 1 on the lower left, you see A, C, T, G, and T, and the C at the first position is the highest, the most that we see is red. So we would call that position a C. And then ultimately that gets turned into that file that shows the sequence of reads in the FASTQ file. Why am I showing you all this biochemistry? Why am I showing you all these different steps? It's because the images in the lower right, we have thrown away since the beginning of sequencing. It's just too big. The machine itself does this computation. We just throw those away. That's not the raw data. On the lower left, the place where we see all the color sequences, those also get thrown away. It's just too big. So we end up ultimately with these raw sequencing reads that are just the letters on the left-hand side of this graphic. And it's important to realize that there were 2 levels of raw data, at least below the sequence FASTQ files, as they're called, that we just threw away and discarded because we didn't think there was enough information in them that was worth keeping the gigantic data. So you can kind of take that idea further and start to think about how could you summarize the data even further? Do we need to keep the entire raw FASTQ file around for every single sample that we have? And so we started thinking about what are summaries that would capture most of the information that we wanted from the sequencing, but wouldn't be so enormous. And so the first stage of this is just to basically align the reads to the human genome. So you can do this alignment. It has to be splicing aware, because remember, some of the reads cover 2 different exons after those 2 exons have been spliced together. But this is a fun computer science problem, and lots of very smart computer scientists have come up with ways to do this kind of alignment, including my colleague Ben Langmead, who's done a bunch of those and was one of my main collaborators on this project. So you align the reads to the position in the genome, and then basically at every position in the genome, we just count the number of reads that cover that position. So here, at the first position, there are 2 reads that cover it. At the second position, there are 6. At the third position, there are 0, and so forth. So you can just count the number of reads covering each position. This is a dramatic reduction in size because we're not keeping every little sequence. We're just counting the number of reads that cover, and most of those counts are 0, so we can compress the data significantly by just looking at this. We also want to keep track of how many reads covered each of the junctions, so we keep track of those as well. And so if we take just those 2 measurements, we can reduce the size of that 3 GB file into a much, much smaller file, a 71 MB file for a large sample, that gives you just this path of like how many reads cover each position in the genome and how many counts of each junction that you have. The cool thing about that is you can reconstruct almost every gene expression measurement that you want to calculate from just those much smaller dimensional measurements. And so the exciting thing is we can reduce size, make the data easier to analyze, and not lose very much information about what's really going on with the underlying gene expression profiles. The question then is, how do we do this at any kind of scale? We can do this for 1 sample at a time. You can do that on a laptop, but imagine that you had to do it at a much larger scale. And the reason why we are interested in much larger scales is because there's more and more data that are available. When we started this project, it actually was not 22,000 samples. We actually started with, I can't remember, it must have been like a couple hundred samples in the very first version of our study, where we're integrating samples across all of the public data sets. In our most recent run, we looked at the Sequence Read Archive, which is a public place where you can deposit sequencing data. We looked for samples that were labeled human, RNA sequencing samples that came from the Illumina technology. And when we did that, we got something like 247,000 samples that were publicly available and that we could potentially include in this analysis. So that's a pretty large number, and now is well beyond what you would do on your own laptop. And so how do you think about how we do this scalable version of the computation? And now, there are different ways to do it. The way that we did it was sort of through massively parallel computing. This is a figure from the paper where we originally designed an approach to basically doing parallel computing on the cloud to calculate this pre-processing step for all of the different samples. We've since used a slightly different architecture. This work is really driven out of Ben Langmead's group, and the most recent person was really Chris Wilkes and Leo Collado Torres and Casper Hansen, who really drove it forward. But there's these sort of a way that you might think about adopting the other trend that's happening, which is the ability for us to get cloud computing. So cloud computing, as you all are probably very well aware, is basically renting computers on the internet. One of the places you can rent computers on the internet is a partner that we work with here a lot at the Hutch, but also worked with a lot at Hopkins, is Amazon Web Services. You can rent computers from these different zones around the world. And so what we've essentially done when we're trying to do a large computation like this is we've turned a biology problem into a computer science problem, which is the alignment problem, which then ultimately becomes an economic problem, which is how do you rent computers cheaply to be able to do a computation of that sufficient scale? The good news is once you've done that computation, you can calculate all these different measurements. You can calculate the gene counts, the exon counts, expressed regions, transcripts, junctions, all of those are sort of computable from these raw sort of just the count of each position and the junction counts. But we want to do that for all 247,000 samples. So how do we do that? And the way to do it is you rent a boatload of computers all at once, and you run this computation across all of them. So this is from actually the Recount 2 project, which is an earlier version of the project. We've since released Recount 3. And this is a plot of the costs to rent computers from Amazon. And you can see here that there is a spike in the spot market. And this spike in the spot market, so this is a really interesting way of renting computers. You basically rent computers by the minute, and you basically bid every minute how much you're going to pay for that computer. And if you underbid everybody, you get to keep the computer, and if you don't, you lose the computer. And so we designed this to be able to run it at a time when very, very few people are using those computers. But we basically changed the global price of renting Amazon computers as we were doing this computation across all of these different samples, because there's so much computation that's being done simultaneously with one collection of data. So it's, again, you've changed a biology problem into a computer science problem into an economic problem, but then you only have to do this pre-processing one time so that every individual person doesn't have to do that processing. Again, for the Recount 2 project, we had about 70,000 human samples. And for the next little bit, I'm going to talk a little bit about annotation. And so I'm going to connect it to this 70,000 samples. But the current version of Recount 3 that's released out now is 247,000. For these samples, we had 10,000 from GTEx, 10,000 from TCGA. These are large-scale projects that are run by the NIH that specifically collect gene expression data among other different data types. And so these are large-scale projects. There's also 50,000 or so Wild West samples just from the SequenceRead archive, not from any individual big-scale study. If you looked at them, many of them are single-cell expression data sets, and you can detect those mostly by looking at those that are observe less than 1,000 junctions expressed. If you look at those that observe more than 20,000 junctions, those tend to be bulk RNA, but these are all labeled, unfortunately, as bulk RNA samples. And so, as with many parts of this project, we're dealing with other people's data. And any of you that have ever dealt with other people's data, you realize that the labeling isn't always exactly what you'd hope it would be. The information isn't always as clear as you would expect. And so part of the challenge is really understanding what exists in these public domain data sets and pulling them together so that there's something that's synthesized and organized so that people can use it. A cool thing about doing this computation at that sort of extensive scale is that we can look across very large collections of data to see how much of human gene expression variation we've actually already observed. So this is kind of a fascinating graphic that came from one of our earliest papers. And it shows time on the x-axis here. So it's over time. And the time is the submission date of a particular gene expression sample. And on the y-axis, we're showing the first time a particular junction is ever observed in any gene expression data collected and made publicly available. And so what you see is these early studies discovered a large number of junctions because they were sort of the first studies to find any kind of sequencing length for the reads. And then over time, there's been a steady accumulation. And then as technologies improve, you see these spikes in the number of reads that or the number of different unique junctions that we've observed. But you can kind of see right around 2013, there was this sort of tailing off. And basically at any given depth, at any given sequencing depth, we started to observe we weren't finding more and more new junctions at any kind of significant rate. The implication of this is it's a good time to go back and re-annotate the human transcriptome because we've sort of observed all of the transcript variability that we would because it's sort of plateauing out in the number of new junctions that we're observing over time. And in fact, a couple of groups did that. They took our data from our Recount project among some other data types, and the NCBI and EMBL created a new transcript set that just came out. You can see the track from Recount here among the data that they were looking at, and they discovered and redefined hundreds of human genes, and it's exciting to see that happen. Basically, like, we've been able to use these big public data to make discoveries like this, where we actually observe a new human gene that we weren't aware of before. We re-annotate human transcriptome now much more empirically than we had before, because we have this sort of scale of data to be able to look across hundreds of thousands of human samples and really observe where the transcript variability is. So that's really exciting that we've been able to sort of get the genomic data in order, and it's been allowed us to do these sorts of things where we're looking at, for example, the sort of what I would call fundamental biology questions. Things like, what are human transcripts? What are human genes? Have we observed them all? As well as re-annotating transcriptomes. That's all great, but it really doesn't connect it to precision oncology, or the question I asked at the beginning, because we don't actually have the clinical data, the metadata, to be able to answer that question. And so even though we can kind of skip many of the steps with this pre-processed data in the Recount project, we still are in for a lot of data cleaning and organization, because those data have come from these public sources where it's pretty hard to know what's what and what the labels are and all that sort of thing. So while we're sort of slowly moving towards reducing the amount of time it takes to do a project like this, without the metadata, it's still very hard to answer questions. And so the Recount project has this component on the top part of the graph on the left, where we're taking sequence data, we're processing it at a huge scale, and we're putting it somewhere where people can access it. But if we want to actually be able to make progress on prognostic signatures, we actually need the metadata as well. So a big effort we've made over the last few years is to not just collect and process the genomic data, but also make sense of the metadata, especially for these public samples. And so even though we have the gene expression data on the left here, if we really want to answer meaningful questions about human biology and gene expression, we're missing this critical gap, which is sort of the information about the labels of the samples, or the metadata. So we don't have those phenotypes that would allow us to link the gene expression to questions that we want to answer. And the challenge, of course, is that for basically everything on the sequence read archive, the phenotype information is really far from complete. So even sort of really standard demographic variables that are collected in these large studies like GTEx or TCGA are just completely absent from the sequence read archive data. They're not collected, or they're collected, but they're collected in very unclear ways. So for example, if you go and pull the field called sex, they have metadata that's available from the sequence read archive. And if you pull the sex field, you get labels with capital F, lowercase female, all the different versions of this for 36,000 or 3,600 of the samples. But then you also get all sorts of other things, not available, not collected, pooled male and female, et cetera, et cetera. So we get about 44,957 of the sequence read archive samples from Recount 2 just didn't have sex information provided. And then 4,700 had information provided, but in a variety of different contexts. So this is challenging if you want to do even really sort of simple analyses. One of the sort of questions that people often ask in these NIH studies is studying sex as a biological variable. And it's very hard to do that if that information is just flat out not being collected, or is being collected in a way that makes it really hard to use. So again, as always, other people's data is amazing, and it's really useful for doing these sorts of analyses, but it's also really challenging. It's not quite as easily used as one would hope. And so one of the things that we worked on for a little while was developing a machine learning approach, and we're sort of on iteration 2 of doing this right now for Recount 3, where we're trying to develop machine learning models to automatically label samples with metadata. And so for the Recount 2 project, we have the advantage of a couple of large, well-annotated studies, the GTEx dataset, which is about 10,000 samples, TCGA, which is another 11,000 or so samples. And then we have the sequence read archive. So we can kind of build interesting training and testing and validation sets. And the good news is for GTEx and for TCGA, the phenotype information is pretty well annotated for a large collection of variables, which means it's possible to build machine learning models to do a lot of this labeling automatically directly from the gene expression data itself, allowing us for us to make a much more complete data set. And so our goal was basically to re-annotate these gene expression data with a number of different variables. I'm going to go through the case where we are looking at labeling with the sex information. We also did this for tissue and sequencing type and a variety of other sort of phenotypes, but we're going to focus on the case of predicting sex. Here, what we did was we divided the data into a training data and a validation data within GTEx. So this is, again, the train where we're going to optimize the predictor, then we'll test accuracy within study. As anybody who's ever analyzed these large scale genomic data will tell you, though, the out of study effects are huge. And so being able to test out of study is really important when you're building a machine learning method. And so we can use TCGA as an out of study validation set or test set. And then ultimately we can go to the sequence read archive to look at how it performs in the Wild West, so to speak, of gene expression data. I'm not going to go into too many details here, just in the interest of time. We developed an incredibly simple gene expression, a method for predicting phenotypes. It basically boiled down to identifying expressed regions or genes that were differentially expressed between the two or multiple categories of variables we were trying to predict, extract those coefficients, and then build a very simple regression model to basically predict sex and other variables. This worked really well for simple cases like sex and tissue. For things like predicting the age of the samples, we're having to rely on more complicated machine learning methods that we're developing right now in the group. The cool thing about this is that for a lot of the variables that we care about, a lot of the demographic variables we care about, these predictions are pretty accurate. So here I'm showing you the prediction of sex. Again, in the training set, we're 99.9% accurate in predicting sex. The validation within GTEx is again very high. And then the thing that was encouraging to us is when you go out of study and look at the TCGA data, there's still relatively high prediction accuracy. We get a little bit lower in the sequence read archive. Again, remember, we get pretty complicated, not totally validated labels in the sequence read archive. So we thought we would examine that a little further and see if our machine learning method was at fault or if the labels were at fault. And so to understand this, the next couple of graphs, I have to explain sort of the plots we would make. So we were looking at the expression of the Y chromosome. So for our predictions of sex for these samples, we would use expression on the Y chromosome. And so we have a couple of different curves here, and these are the expression levels, a density plot of the expression levels for reported and predicted. In this graph, it shows it like it should, which is what we expect, at least, is that the predicted female and the reported female samples have low expression on the Y chromosome. And so the density plots are over here on the left, and the reported male and predicted male samples have higher levels of expression on the Y chromosome, and you can see the density plots on the right. But when you actually go look at some of these studies, you of course see something that's a little bit different. So for example, in this study here on the left-hand side, SRP 040547, you see that the predicted males have a high Y chromosome expression, but the reported males have a low Y chromosome expression. So we think in this case, they didn't label the males samples correctly. Here in this example, you see that there's like a lot of carryover in the expression of the Y and chromosome for the reported females and the reported males. So we think that there might just be some labeling issues in this one. And then over here on this one, the predicted male and the reported male, again, you see the predicted works pretty well, and the predicted female works pretty well, but the reported doesn't seem to work. And so we saw these examples. We also saw examples where the male and female labels were switched. So in general, we're not really sure that the predictor is doing worse, but we are unsure that the observed labels that we get in the metadata really accurately reflect what's going on in the underlying data. So again, we have this sort of other people's data problem, but in this case, it's really quite fixable. We can predict a lot of these demographic variables directly from the gene expression data themselves. Even for things as complicated as tissue, we usually get high 90% accuracy, even with pretty simple prediction models to get the tissue labels for all of the sequence read archive data. So the cool thing is that using those machine learning methods, we can now get phenotypes for most of the sequence read archive data, which allows us to answer a much broader collection of questions about human biology and gene expression using these well annotated and commonly processed samples that come from a broad collection of studies that cover all sorts of different biological questions that you might be interested in. So now that we have both the pre-processed genomic data as well as the clean and predicted metadata, we can start going back to some of the questions we originally started with. The one thing that I'm excited about, that the preprint is just coming out this week, so stay tuned. is we also collect the sequence, the gene expression profiles for both the reference DNA sequence, but also for alternative sequences for DNA variants. And so using that data, we've been able to develop algorithms for basically counting how many alleles, how many of the gene expression measurements cover the reference SNP allele versus the alternative allele. And so using that and some very simple machine learning techniques, we've been able to genotype most of these samples. And so we have both genotypes and gene expression measurements for all of these hundreds of thousands of samples, which is quite exciting. I'm not going to talk too much about this today. That's coming out soon. Was so excited about it. I wanted to show you that now we have phenotype information, gene expression information, and genetic information on hundreds of thousands of human samples. And so the way you do that really is just basically calculating the difference in counts. That's the y-axis. The m-axis is an MA plot for those of you that have seen it before. It's sort of the difference between the counts for the reference and the counts for alternative, and you plot it on the x-axis versus just the overall abundance of that particular SNP. And you can see that you get these decision boundaries that show whether it should be called homozygous reference, homozygous alternative, or heterozygous, depending on which of these regions this MA plot falls into. It's a really simple machine learning that seems to work really well across all of these sequence read archive samples. And so we get, again, very, very high accuracy. On the left is calling all the samples major allele only, which does very well on the major allele, but terrible on the minor allele. On the right, we get very high accuracy on both the major and minor allele if we use this kind of simple machine learning model and are able to genotype a large number of samples. Yes. And there's a good question that's been asked by Seth. If we can't trust researchers to put the metadata in, how much faith can we have in the metadata? Yes. So I would say that metadata that you find in the sequence read archive is you should view with some skepticism. For these large scale studies that the NIH funds, one of the things that they do is they're very careful about metadata collection. And so we've been using those as training sets. Ultimately, though, we need more cases where the metadata is really put together in a way that is as systematic as things like GTEx and TCGA if we're going to be doing this. So yeah, I'm less worried about these machine learning models, but they're somewhat limited to only the variables that are collected in GTEx and TCGA as training sets, to be totally straightforward about that. All right. I'm trying to keep an eye on the questions as I go, so if people want to stop me as we go. All right. And then I'm going to move on to the next step of this sort of process, which is sort of the analysis step. So you've got the metadata hopefully labeled. You've got now maybe some genotype data. Now we can go back and answer the question we originally started with maybe a little bit quicker, because we basically can take data off the shelf and answer this question. And so going back to what gene expression patterns are prognostic of colorectal cancer metastasis, this question we can go and pull out, in fact, the original data set that they used to answer this question from our process data, and we can go back and do a little bit of reanalysis. And so in this data set, they have 18 samples. They have healthy colon samples, normal colon NC. They have primary cancer PC samples, which are, again, sampled from the colon, from the cancer tumor, and then liver metastasis samples that were sampled from the liver, 6 of each type of sample when they were building their gene expression profiles. And so we can go and do the comparisons that they did in their study, where you compare the metastatic samples to the primary cancer on the left, and the normal colon to the primary cancer, or the non-cancerous to the primary cancer on the right. And we can look at how many genes they found differentially expressed in the green plots and how many we found in the pink plots. Some of this is just due to differences in the types of analysis that we did, and not necessarily due to any big difference in biology. And then we can start doing some more interesting analyses. So for example, we can condition on sex, because they didn't do that in their initial analysis. They didn't make that data available in the metadata, but we have a prediction for it. So we can use that and see if we condition on sex, if we see a difference in the analysis. And when we condition on sex, we don't see much difference. But I'm going to talk a little bit about what happens when we condition on tissue. And so to be able to do that, I want to talk a little bit about how you compare two different analyses to see if they're the same or if they're different. And the way that people do that is with something called a concordance at the top plot. So when you do two different differential expression analyses, you're basically looking for which genes are different between primary cancer and normal tissue. And in that analysis, you do say there's 20,000 genes, you rank them from most differentially expressed to least by something like a p-value or a test statistic. And you can do that for different analyses and then compare them by basically ordering all the genes by their results from analysis A, and then seeing what the order is for analysis B of how many genes among analysis B are the same as those for analysis A. So for the top 10 for analysis A on the x-axis, we count what fraction are also in the top 10 for analysis B. If the analyses are identical, it should be 10. So you basically get a horizontal or a sort of 45-degree line if the two analyses are identical, because basically the same top 10, the same top 20, the same top 30, the same top 40. In practice, what you actually observe for two analyses that are very close to each other is that the top results are basically the same and are more or less follow along the 45-degree line. And then you start to see some differences at less significant genes. If the two are very different from it, if the two analyses are very different from each other, you see an immediate divergence from this 45-degree line because the top genes for analysis A are not the same as the top genes for analysis B, and so they give you different results. So why did I do that little diversion? Because I wanted to talk a little bit about what we found to be the crux of this particular prognostic signature, which is, so if you condition on sex, there's no real differences. You can see that the analyses are basically the same. If you do the normal versus the primary, and if you're doing metastatic versus primary, and you do that analysis condition on sex or not condition on sex, you basically get the same analysis. But what happens when you condition on tissue? So again, they didn't produce the tissue for the different samples other than the original labels, but we have predicted tissue for each of the samples. And so when you condition on tissue, you see very few differentially expressed genes, and it's a very different analysis. And so what's going on? If you condition on tissue, then the metastatic versus primary cancer with covariates on the y-axis, and the metastatic versus primary cancer without covariates are very different analyses. So basically, when you look at the metastatic versus primary, the prognostic signature they were originally using, you get a totally different set of results when you condition on tissue. So why is that happening? So our hypothesis was that the loss of concordance between these two analyses suggests that the differential expression is actually detecting tissue differences and not cancer-related changes. And so the good news is we can actually do a comparison because we have all of these other data sets that have been processed on the same processing pipeline. So we can look at a comparison of healthy liver and colon samples from the GTEx study and see if we get the same results as we get from comparing the primary cancer to the metastatic cancer, because that would suggest that primary versus metastatic is not predicting the cancer status, it's actually predicting the tissue type. And so our hypothesis is that the metastatic versus the primary cancer results should be most similar to the GTEx colon versus liver results, because we're basically think we're predicting tissue versus predicting cancer status. And in fact, that's what we observed. So if you compare the GTEx MC-PC analysis in Kim et al. And the GTEx liver versus colon, you actually get much more concordant results than if you do the same, just for reference comparisons, normal versus primary cancer, you don't get the concordance between the Kim et al. Study and the liver versus colon analysis. So in other words, sort of validated the hypothesis that it's likely that the differences between the MC and PC samples are not cancer differences, but they're actually tissue differences. And so the signature is not prognostic of metastasis, it's actually just predicting tissue. And so this ultimately would led us to not use this signature to pursue it further, sort of for the precision oncology efforts that we were doing with those collaborators. And so I think that the interesting thing about that analysis that I just showed you is a couple of things. One is we were able to quickly look at a question of interest without having to process the data and collect them and integrate them. Because all the data were processed on a common pipeline, we could do this meta comparison across the GTEx tissues. And all of that was possible with just a few lines of code in R for us to be able to do this analysis. So that was quite exciting. And while that process works once, how do we actually scale it to more people is something that we've been thinking a lot about in my group. And the first step is really thinking about, okay, if you do an analysis once, you just write the analysis. If you want other people to do the analysis, you need to create software so that they can do the analysis. This is analogous to what we're doing here at the Fred Hutch Data Science Lab, where we're building data resources that people can use, but we're also building the tools that they can use to analyze that data. So we did that in a few different ways. We built a study explorer that's at this URL where you can go and look at each of the studies that we've collected and all of the 240,000 human samples and the 350,000 or so mouse samples, and sort of look through those different studies and find gene expression measurements you might be interested in. We've also built a collection of bioconductor packages. The Recount3 package will let you programmatically access all of the gene expression I just talked about and more. The Snaptron package, which allows you to look at individual splice junctions across many thousands of samples. And then the DASPER package, which looks at the relative frequency of different splicing junctions across different samples to be able to sort of analyze them collectively. And then there's one more step, though, because most people might get excited about this, but might not have the background to be able to analyze the data. So again, similar to what we're doing at the Fred Hutch Data Science Lab, we want to layer on a level of training to be able to get everybody to be able to use these data. And so we've developed a number of large-scale online programs like this genomic data science specialization, which essentially allow people to learn about data science and genomic data science at a scale that wasn't possible when we would do it locally. And so we're now about to release some new courses in AI that I think are pretty exciting out of the Fred Hutch here, that similarly try to level set everybody at a new level with these new technologies when they come out. And then we also run workshops. We run them in person at these various different meetings. We continue to do that, though. In the New World Order, the in-person workshop is suddenly kind of how much of that is going to happen in the future is sort of anybody's guess. With that, I'd just like to thank all of the different people that have participated. This is a big project that has spanned basically a decade of my career. My closest collaborators have been Ben Langmead and Casper Hansen. Avi and Chris have been big proponents and leaders in the sort of processing of the data at scale. And then Shannon was most of the metadata prediction that you saw. Leo has been a major Recount bioconductor package developer, as well as a core participant in all of these projects. Afrooz and Chris are doing the genotyping information, and then Alyssa Frazee was the first student way back in the day that sort of kicked all this off. So anything cool that you've seen here is due to one of these groups. Oh, and Andrew Jaffe, who's also been kind of a core collaborator since the beginning. And anything that seems wrong or messed up is probably due to me. Again, I just wanted to call to your attention these sort of places where you can stay in touch with us. We're excited about interacting with you a lot more. And I wanted to thank you all for your attention and thank you for taking the time to invite me here today. So thanks very much and happy to answer questions if you have them.